I'm just going to talk about sustainable harnessing of bioactives from food waste. And I suppose this is one of the research areas that I'm involved with. It's not the only one I'm involved with, but it's probably the one that I'm most involved with. So when you invited me to give the talk, you asked me to sort of address it in a particular manner. And one of the things you asked me to do was to look at the challenges. So I think we're probably all pretty much aware of what the challenges are within the area of waste. They are huge. I mean, in Europe alone, we generate 90 million tons of food waste. And actually, a lot of waste also arises from the harvesting of crops as well, which could also serve perhaps as a source of, or as a resource. The UN has said lost or wasted food products across the world costs approximately $850 billion. And in fact, waste of the resources contributes to annual global greenhouse gas emissions. So it's a huge problem. I think we're only at the very start of trying to address it. And I suppose this morning I'm just going to try and talk about the way in which I am contributing towards addressing of this challenge. So what could our possible response for? So on the left-hand side there, you can see the amount of waste from food processing and the amount of waste from the harvesting of crops that is generated. And I suppose the challenge or the response that we're going to try and to address this is to convert it into something of use. So it could be converted into energy. It could be converted into some sort of chemical or material or starting material for the manufacture of something. And the area then, I suppose, that I've worked the most in is to try and get a food ingredient from this type of waste. And then finally, it can go all the way towards a pharmaceutical that we could transform or harvest a pharmaceutical from the waste. And as you can see, as we go from energy to pharma, the value of the component that we're harnessing from the waste is increasing. So, and again, just to emphasize the opportunity that waste might represent, if we look at, first of all, in the first graph there, we're just looking at the total amount of waste generated. Europe is right up there. North America generates more waste, but Europe generates an awful lot of waste. If you then look at how much different types of commodities generate waste, so in figure 4 there, that's looking at the amount of waste in different areas in the world, and again you can see that it's quite high, and that roots and tubers generate a lot of waste. A lot of roots and tubers are actually discarded. I mean, up to 50 or 60% in some cases are discarded during processing, or perhaps thrown away in domestic use, etc. If you contrast that with milk and dairy production, the levels of wastage are much lower, and that's because historically there has been a recognition of the potential of the waste from dairy processing, and we all are very aware of the different projects and the different commodities that have been generated and valorized by the dairy industry. And of course, within Ireland, we're the biggest exporter of infant formula, which is generated in part at least from dairy waste that would have been discarded before. So you can see the opportunity that it presents, and also then the decrease in environmental damage that would result if we could reduce the amount of waste that is generated from fruits and vegetables, from plant food waste, from an environmental and also from a capital generation perspective. So I've just put this slide up here to give you an example of the type of waste that I've worked on. And I suppose these are examples of waste and byproducts that are commonly generated by industries that are fairly well established in Ireland. We've got apple pomace, which is what's left over after the pressing of apples, and we've got a big cider industry in Ireland that would generate a lot of apple pomace. We've got potato peel. We've got a big potato processing industry as well, but that would generate a lot of potato peel. We've got a big mushroom producing industry as well. And the mushroom stalks from that industry are often discarded. We've got a big brewing industry, and the biggest waste generated from that would be the spent grain after the fermentation process. I've concluded another two here, which aren't really sort of byproducts, but they are something that I've worked on before. So there are a lot of underutilized plants, and by that I mean perhaps what you would call weeds that could contain some compounds that would be of value. And I think there's a growing recognition of seaweeds as a possible source of bioactives, and I've worked a little bit on that area as well. What kind of compounds have I worked on? Well, I suppose the ubiquitous one, the one that I've worked on the most, would be polyphenols. So polyphenols are secondary metabolites produced as defense molecules in a lot of plants. I would give an example there of caffeic acid, which is one you might find in potato peel. I've also given an example of a condensed polyphenol. They're known as fluorotannins, that you'll find in seaweed. So, you can find them in unexpected places as well. The second group of compounds there are glycoalkaloids, and that I've worked on them a little bit. I've mentioned them here on this slide because they've come back to haunt me recently in a new project that I'm involved with. But I was interested in them previously before as a source of a phytopharmaceutical, we called it. Basically, it was the starting point for the manufacture of an anti-cancer drug. And then we've got beta-glucans as well, which I've included on the slide because they're an example of a bioactives that have an EFSA validated health claim associated with them. And then just to again emphasize the opportunity, if you look at the global market for polyphenols, it's 1.28 billion in 2018, and that's expected to increase. The global market for beta glucans is 313 million, and again that's expected to increase up to 2025. So there's a need for these molecules. These molecules are present in waste. So the next challenge is really to try and get them out. And that is quite a challenge because what you're trying to do a lot of the time in this area is you're contributing towards sustainability. You're also, I suppose, generating income for people. But often the way in which you extract, or the way in which you could extract the molecules doesn't contribute towards sustainability. It's often quite unsustainable. For example, it generates a lot of waste solvents. It's often very energy inefficient. So the recovery of the molecules from the waste sometimes creates as many problems as the generation of the waste itself. So what we need are more sustainable, more cost-effective technologies that will allow us to harvest the compounds that we are after using less energy, therefore less expensive, and less solvents as well. So there is absolutely no point in saying we can make this great contribution towards the sustainability unless we do that in a sustainable fashion. If we can do this, if we can help with, or if we can harness plant food waste in particular as a source of bioactives, what impact can we have? So we can develop new value chains. So for higher added value products. So if we go back to that second slide, you can see you can generate chemicals, you can generate food ingredients, etc. You're going to improve environmental performance if you can do it in a sustainable manner and a cost-efficient manner. And you can then develop and validate new products with higher values than the current raw materials. And of course, you're going to increase your sustainability score compared to, for example, a fossil fuels-based counterpart. And as I said, there's a market there, so we're meeting a clear market demand if we can do this. Now, if we go back to, I suppose, the niche area that I'm most involved with is this area of extraction. So the area of extraction is very important. I think I've emphasized that already, because if you can't sustainably extract the bioactives that you are after, you're not really going to, it's not going to be cost effective. It's not going to be environmentally friendly. So there are technologies available, and the two that I've worked on the most are ultrasound assisted extraction, pulse electric field assisted extraction, and they contribute towards the sustainability, the cost effectiveness of the harnessing of bioactives, particularly from plant food waste, because what they can do is they can break cell walls. So if you can break cell walls using less energy than would be required, say for example, in a mechanical fashion, or using a solvent to do so, you can release then the components that you're after, that you're trying to harness from the and harvest from the waste. So both ultrasound and pulse electric fields, they can break cell walls, and because you can break cell walls, you can use less solvents, you can use less energy, etc., in order to extract the bioactives out of the food waste. So you're using less energy, so it's low energy, you're increasing sustainability. Now, everything has its advantages and disadvantages. In comparison to the normal method that would be used, which would just be solid liquid extraction, you'd have a much higher capital cost if you were to use a technology such as this in order to harness food waste for bioactives. And that's because, you know, you have to buy specialized equipment in order to be able to do this. But having said that, these technologies, they are scalable. So they have been, and they can be scaled up to treat plant food waste in order for you to harvest bioactives from them. So I think I've talked a lot about the opportunity, the challenges, etc. What I wanted to do in the next couple of slides was just to talk about how do you address this challenge, and also what collaborators we need to address the challenge. And it also gives me the opportunity to talk about a project that I'm involved with. So I'm involved with a bio-based industry project called Agrimax, and even the numbers on this slide, I think, probably illustrate the challenge that harnessing plant and cereal waste as a source of bioactive and also energy in the case of Agrimax. There's a huge number of partners involved in this. There's partners from all over Europe involved in this. It's fairly big money, so it's 15 million euro. But I suppose what's most important about Agrimax is the multidisciplinary nature of it. We have people with lots of different skills, all working in a collaborative fashion to try and harness bioactives. In my case, I'm most involved in looking at potato peel as a source of both potato peel and also potato press juice as a source of protein in terms of the press juice, and also then the peel as a source of polyphenols. But I'm not working on my own in that. I have people who have been involved in life cycle analysis, of people who are involved in scale up, etc. And we're all working together in very much in a collaborative fashion to address this challenge. And I think that's at the core of being able to address this challenge. It does require lots and lots of different skills in order for us to be able to do this effectively. And this is just an example of one of the things I was involved with. One of the rest raw materials that we're interested in Agrimax is known as olive pumice. And olive pumice contains another actually EFSA validated bioactive known as hydroxytyrosol. And hydroxytyrosol can reduce oxidation of cholesterol, and it has a health claim associated with that. And what we've done is we've developed an ultrasound assisted method to recover a lot more of hydroxytyrosol from the olive pumice than the conventional method. So the first bar there where it says solid liquid, that would be the conventional method, just putting a solvent. In the case of hydroxytyrosol, it would be an ethanol water mix in contact with the olive pumice and extracting then the hydroxytyrosol into the solvent. However, we found that if we use ultrasound assisted extraction, I can't specify the conditions because they're looking into protecting it. But if we use a particular set of ultrasound conditions, we can harness a lot more of hydroxytyrosol from olive pumice. So with respect to this, I was involved in the development of the extraction method, but also then in characterization and in fact of the hydroxytyrosol using HPLC here in UCD. Another thing I alluded to earlier on, I was talking about the different molecules that we are involved with. As I said, one of the things that I'm involved with, most involved with in Agrimax is potato protein, and they're trying to recover potato protein from potato press juice, which would normally be a waste. However, what they found is that one of the molecules that I was interested in before, glycoalkaloids, are being co-extracted within the potato protein, and this is a problem because glycoalkaloids they're toxic above a certain level, so you don't want them in your potato protein. So I did warn them before they looked at this. This might be the case because I was already aware that they could be co-extracted with polyphenols when I was trying to harvest polyphenols from the potato peel. However, we were able to develop a way to sort of detoxify the potato protein. So basically, we've used a fairly low concentration acetic acid extract, which harvests the glycoalkaloids from the potato protein and gets the glycoalkaloid level down to a level below that which is toxic. That means that the potato protein that we're extracting is still, suitable for use as a food ingredient. But it is another example of one of the things that another problem that you could come across when trying to harvest bioactives, particularly from plant food waste. You may co-extract and concentrate other compounds that you don't want that could be, in the case of glycoalkaloids, they are toxic. Then all I just wanted to show you is that Agrimax is a demonstration project, and what we did in Agrimax is we developed methods for extraction of the bioactives, first of all at lab scale, but they are now being implemented in an actual pilot plant. So this is an example of the design of the pilot plant. So I inputted into the design of the pilot plant for polyphenol extraction. This particular workflow it has been designed both for extracting polyphenols from potato peel, but also for extracting polyphenols from, actually hydroxytyrosol, which is the polyphenol, from olive pumice. And it's very gratifying when you see some of the work that you've done being actually put into action. So on this slide here, I've just shown some examples of of people actually putting together the pilot plant that we designed. See the top right hand corner, that's a big solid liquid extraction unit that's used in the polyphenol extraction step that I helped design for potato peel and also for olive pumice. And I suppose one of the things I think we always have to be as realistic as we can, and I think there are still big obstacles in our way with regard to this particular area for plant food waste. One of the biggest one is the homogeneity of the raw material. You need lots of raw material in order to have enough to get enough of the bioactive that you're after, and you may not always be getting the same thing. Sometimes you might be getting for example, if we took potato peel, you might be getting a potato peel that has a lot of polyphenols in it because it's coming from one particular cultivar. At another time of the year, they might use a different cultivar and you'll get less polyphenols. So if you're trying to extract a bioactive and turn it into something that's marketable, you want to always be getting the same amount out. Also, I said, you need a lot of the raw material in order to make this a viable industry, and there aren't that many industries that generate enough in the one place. So it's quite difficult to carry out this based on the waste generated from one particular company, for example. So the logistics of carrying out it can be difficult. And in fact, within the Agrimax project, they've recognized this, and they have a work package that's looking at developing ways in which processors could bring all their waste to a particular place, and such that then we'd have enough in order for the process of harvesting the bioactives to be viable. One of the things I came across that was a problem with regard to the Agrimax project is that I developed an extraction process for polyphenols from potato peel, but using an ethanol water mix as the solvent. However, while ethanol water is that is the best way of getting out, and it is food friendly. In other cases, the solvents that extract the bioactives to the highest degree are not the ones that are food friendly. So because we're looking at a food ingredient, we're always going to be limited to food friendly solvents. So that would be usually water or an ethanol water mix. Co-extracted toxicants, of course, again, I showed you in a particular example of that if you're concentrating your bioactive, your target molecule, sometimes you're also concentrating a toxicant as well. The valorization process. So what do you do if you extract a bioactive from potato peel? What do you do with the potato peel that's left over after you've done the extraction? Now, within Agrimax, we are trying to address this. We're looking at always examining what's left over after we've harvested a particular bioactive and trying to use that as the source of something else, because it has to be, you have to address this in a circular fashion really to make it as sustainable as possible. Again, the sustainability of the valorization process must always be, can always be an obstacle. It's quite difficult to get everything out, but also ensure that you're doing that in a sustainable fashion. And again, the primary producers and the processors, they're very much aware actually of the value of the waste that they're generating, and they want to be able to use it or to sell it as a source of a particular valuable component. However, they're not really aware of the technologies and the methods that are required for them to be able to do this. You did also ask me to look at funding sources, and what I've done here basically is just give a history of the type of funding sources that I have used. DAF, of course, through the Food Institutional Research Measure would be one, and they were actually a little bit ahead of the game back in 2000 when they funded a project of mine on this, looking at waste and byproducts from food and vegetable processing as a source of compounds. They also funded a big project of mine called the IPFN, the Irish Phytofood Network, and within that we were also looking at lots of different sources of plants and foods or lots of different raw materials such as plants and foods as sources of functional molecules. Back in 2008, then a firm also funded a project of mine, and this was the one where we were looking at potato peel waste as a source of glycoalkalies to use as a phytopharmaceutical. They've also kept on supporting me up till 2011, where myself and Jim and Dilla Bray in Charcas, we're looking at a project which is directly using some of these novel technologies as a way in which we can sustainably harvest bioactives from plant food waste streams. And then the final one, or the second last one here, is the one I've already mentioned, the Agrimax project, and that's a bio-based industry. So this is money coming from Europe to fund this particular area. And then right up to 2018, I'm involved in Beacon, and in Beacon then we are using some of those sustainable technologies to address the wider bioeconomy. So there are lots of other funding agencies for this. Well, I suppose what I would say is that it is an area in which you can get funding. It is a fairly well funded area, because people have recognized the need for sustainability, the need for reducing carbon footprints, etc., etc. So it is a good area for funding. And that's it. Thank you. I hope I haven't gone on too long. That's all I have to say for this morning.